All right, hello everyone. This is the Target 3A notes. There's going to be another set of notes after this, but this is the first step. And during these notes, we are going to focus on meiosis and mitosis, what they are, what happens during each of them, and then you will be able to use these notes on the upcoming practice assignments and quizzes. Just as a little heads up, this target is very vocab heavy, so we're going to do a lot of vocabulary practice. You should also have you should also have taken the DNA practice assignment as well. And gotten 100% on that, because in order to understand meiosis and mitosis, you have to know DNA. You have to understand that DNA is what makes us who we are, to understand what's going on during mitosis and meiosis. So for this target, by the end of it, this is what I want you to be able to do. This is your learning target. So by the end, you should be able to create a model of meiosis to demonstrate how characteristics of one generation are passed to the next with variation. So this is what I'm going to hopefully see that you can do by the end of this module. All right, so we're going to start out by watching a video of what mitosis is, because in order to understand meiosis, you have to understand mitosis first. So this is our overview of what mitosis is, and then we are going to look at what meiosis is, and we're going to kind of compare and contrast those two. But we're going to dive really deeply into what meiosis is, because that is the target. We're talking about meiosis. So here's our video about what mitosis is and why our body does mitosis.
Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of it, of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes. Very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes, just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached, and we're counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there. And it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells, and in the human body they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, so the important thing to know for this video is that you do not need to have memorized all of the steps. You don't need to know telophase versus prophase and things like that. The whole goal of mitosis is to go from one cell to two identical cells. So that just walked you through that process. How do we go from one cell to two identical cells? Because that's the entire goal of mitosis. So that way we can do things like grow larger. We can do things like replace our cells. In order to do those things, in order to grow, in order to replace older damaged cells, you have to have new cells. And in order to get those new cells, we have to start from one cell and make an identical cell. So that is kind of your overview of mitosis. And we're going to move on next to talk about meiosis. Sorry, I spoke too soon. We're not moving on to meiosis quite yet. We are going to take some notes over mitosis. So these are two of the things that you need to write down in your notes about mitosis. So I would start this out by putting target 3A, mitosis and meiosis, at the top of your paper. And then we can start with mitosis notes. So what is our definition of mitosis? This is the process in which a cell divides from one cell into two identical daughter cells. So we're going from one cell to two cells. Those cells look exactly the same. They have the exact same stuff in them. They are identical in every single way. So if we have like a lung cell that goes through mitosis, it's going to create another lung cell. If we have a skin cell that goes through mitosis, it's going to create an identical replica of a skin cell. So why do we do this? We need to go from one cell to two identical cells. Our body needs to be able to create more cells. When our lung cells get damaged, because maybe we inhale some smoke or something, some of those cells are going to get damaged, they're going to die off. We need to be able to replace those damaged cells by going through the process of mitosis. In order to grow larger, so obviously we are not the same size as when we were a baby, in order to grow larger, we have to create more cells. So in order to create more cells, we need to go through this process of mitosis where we are creating identical cells, which allows us to grow. So if you have not written these things down, I would get these written down. This picture down here is helpful. It shows how we go from one cell to two identical cells. You don't really need to draw the stuff that's inside of it. The biggest thing that you need to know is that we are going from one cell to two identical cells. All right, now that we have talked about mitosis, we need to talk about meiosis because we are going to compare and contrast the two. And meiosis is the big thing that we are going to focus on for this target. So understanding mitosis helps you to understand meiosis, because if you don't understand mitosis, you don't really understand meiosis. It's not the main thing for this target. The main thing is meiosis, but we have to have that background knowledge to understand what's going on here. So we're going to watch a video very similar to mitosis, but there are very big differences in meiosis. So for this video, you don't need to take notes. We're going to walk through the steps of meiosis together, but this is like an overview, kind of an introduction to meiosis and what is happening.
Have you ever wondered how two siblings can have the same mom and dad and still look so different? Well, today we're going to talk about a process that makes that possible, a process called meiosis, not to be confused with mitosis, which sounds unfortunately similar. Mitosis makes identical body cells, like your skin cells and stomach cells. Recall from our mitosis clip that since it makes identical body cells, mitosis is important for growth and for repair of damage, or to replace worn out cells, but not meiosis. Meiosis is a process that contributes to genetic variety. Meiosis also doesn't make body cells. It makes sperm and egg cells, otherwise known as gametes, the fancier word. You might recall that humans have 46 chromosomes. That's how many chromosomes most body cells in your body have. But there are some human cells that don't have 46 chromosomes. Human sperm cells and egg cells have 23 chromosomes. Why the number difference? Well, if a sperm cell has 23 chromosomes and an egg cell has 23 chromosomes, when they come together, that makes 46 chromosomes. That will allow a newly formed fertilized egg to develop into a human. Meiosis is what we call a reduction division because you have a starting cell that has 46 chromosomes, and your ending cell, the sperm and egg cells, have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. Actually, this also happens before mitosis. It's the stage known as interphase. If you remember interphase, it's when the cell is growing, it's replicating its DNA, it's carrying out cell processes, and just like mitosis, interphase happens before meiosis is even going to start. So the starting cell has 46 chromosomes, and you have to duplicate those chromosomes in interphase before meiosis starts. That basically means you are duplicating your DNA, since chromosomes are made of DNA and protein. Ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, because the sister chromatids are still attached, and we are actually counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture. But you went from 46 to 92 chromatids. Little tricky there. We have a detailed video that explains these chromosome numbers before and after replicating in interphase that can be useful for meiosis. Okay, so interphase checklist done. Now we can move into meiosis. You might remember the mitosis stages PMAT. The P was for prophase, the M was for metaphase, the A for anaphase, and the T for telophase. And the good news is that in meiosis, you still use those terms. But because meiosis is actually a reduction division, you're going from 46 chromosomes to 23, which means you actually divide twice. So instead of mitosis, where you divide one time and do PMAT one time, in meiosis you're going to divide twice, and therefore do PMAT twice. And because of this, in meiosis, you put numbers after the phases to indicate whether you're in the first division or the second division. So let's dive right in. So let's start with the very first step, prophase 1. One thing I like to remember about prophase is pro, this actually means before, and it kind of helps you remember that it comes before all the other stages start. This is where the chromosomes are going to condense and thicken. They are actually going to line up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. They're going to match up. It is during this prophase 1 that this amazing process occurs, called crossing over. Now I know crossing over probably sounds like something very different, but this is actually a really awesome process because this is when the chromosomes, they're lined up in homologous pairs, and they have this way that they can transfer their genetic information and exchange it between each other. It's kind of like these chromosomes flop over each other and they do a little genetic information exchange here. It makes for what we call recombinant chromosomes, which can eventually contribute to the variety that we were mentioning that siblings can have even when they have the same parents. More about that later. Now we move into metaphase 1. In metaphase 1, think of the M as standing for middle. The chromosomes are now going to be in the middle of the cell. It's a little bit different though from mitosis because these chromosomes are going to be in pairs in the middle of the cell. So it's not a single file line, they're in pairs in the middle. Now during anaphase 1, think of A for away because the chromosomes are going to be pulled away by the spindle fibers. Then we end with telophase 1, where you have two newly formed nuclei, and it becomes obvious that you're going to end meiosis 1 with two new cells. Cytokinesis follows with splitting the cytoplasm. But we're not done yet. On to meiosis 2. The very first step in meiosis 2 is prophase 2. It's not going to be nearly as eventful as it was in prophase 1 though, because they're not going to have homologous pairs of chromosomes. They also are not going to have this amazing process called crossing over that doesn't happen again in prophase 2. You have your chromosomes, and the spindles are starting to form like they did in prophase 1, but prophase 2 is just not as eventful of having that process of crossing over. In metaphase 2, remember, think M for middle, the chromosomes are going to line up in the middle. This time, though, they are in a single file line. They are not in pairs like they were in metaphase 1. Anaphase 2, remember A for away, but this time it's the chromatids that are going to be pulled away by the spindle fibers. In telophase 2, you can see the nuclei reforming, and you can also see that the two cells have divided. There's going to be 4 cells forming. Cytokinesis will follow to completely split the cytoplasm. Now, keep in mind that meiosis in males produces sperm cells, and in females it produces egg cells. Because of independent assortment and also crossing over, you're going to have variety. For example, in a male, the 4 sperm cells that are produced each time, they are all different from each other. They're also different from the starting cell because the starting cell had 46 chromosomes. and the ending cells only had 23. So they are not identical to the original, and they are not identical to each other. This is going to lead to variety, a reason why two siblings with the same parents can look different from each other. They still developed from a unique egg and a unique sperm cell that came together. One last thing to think about. Scientists are often looking into the process of meiosis because sometimes the chromosomes don't separate correctly. It's called non-disjunction, when a cell can receive too many or too few chromosomes in the separation. This contributes to some genetic disorders, which is something scientists continue to study. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, so that is kind of our overview of meiosis. This might be a good video to go back and rewatch if after we're done taking notes. You're still pretty confused. If after you are done with the practice assignments and you're about to take the assessment, you want just another review, because this kind of goes over all of the parts of meiosis and what we are going to talk about. All right, so now we're going to start with our meiosis notes. So this is where you need to start writing some stuff down. For our definition of meiosis, it's the process in which we go from one cell to 4 haploid daughter cells. So that's some of our vocabulary terms. You're going to have a vocabulary practice here in a second, but when we're talking about haploid, we have half of the number of chromosomes that we originally started with. And when we're talking about daughter cells, these are just the cells that result from cell division. So when a cell splits in half, it creates daughter cells. That's what we call them. There are no son cells. We just name them daughter cells. It's just the cells that result from cell division when a cell goes from 1 cell to 2 cells or from 2 cells to 4 cells. So what is the purpose of meiosis? Why does our body do it? It's to go from 1 cell to 4 haploid daughter cells so that an organism can reproduce. So we start out with 1 cell, we end with 4 cells, and those cells are going to have half the number of chromosomes in them. And so the reason they have half the number of chromosomes is because we have mom and we have dad. So dad has sperm cells, mom has egg cells. If they had the total number of chromosomes, so for humans, if they had 46 chromosomes and they combine together, they're going to have 92 chromosomes. We don't want humans walking around with 92 chromosomes. We want humans to all have the same number of chromosomes. We want them all to have 46 chromosomes. So when we have a sperm cell and when we have an egg cell and when we combine those cells need to have half of the number, or 23, so that when they combine together, it will equal 46. So that's why when we start out with a cell that has 46 chromosomes, we end with 4 cells that have 23 chromosomes, so that when they pair up with their match, so when an egg pairs with a sperm, when a sperm pairs with an egg, those 23 chromosomes will combine and they will give us 46 chromosomes like humans should have. So we will kind of get into that a little bit more, but that is our overview of meiosis, what it is and why we do that. So we're going to break down meiosis into its steps. So this is the first step. I don't know why it doesn't say step 1, but it should say step 1 at the top. So when you're writing this down for your notes, you need to write meiosis step 1. So this is our somatic cell. It's just a normal cell. And it contains 2 pairs of chromosomes. So like I said, normally humans contain 46 chromosomes or 23 pairs of chromosomes, but we are not going to write out all of those chromosomes. That would be a lot. So as you are drawing these pictures, my suggestion would be to either get 2 different colors to represent the 2 different chromosomes, one from mom, one from dad. Kind of like I have here, we see that there's like a darker bluish black chromosome, and then there's like a green chromosome. It might be a little bit difficult to see. Then there's this little black dot in the corner. That doesn't mean anything. That was just accidentally there. What we're focusing on is the cell itself and the chromosome pairs that are in the cell. So we can see that there are 4 total chromosomes, 2 blue of them, 2 green of them. 2 of them are small, 2 of them are large. So these are our 4 chromosomes. We have the blue ones that would probably come from dad, and we have the green ones that would come from mom. So in each set of chromosomes, so we have 2 small ones, one from dad, one from mom. We have 2 large ones, one from dad, one from mom. So as you are drawing this, I would get 2 different colors, or I would maybe shade one of your chromosomes in and leave the other one open, so that way you can tell the difference, because it's important that we can tell the difference between the chromosomes from mom and the chromosomes from dad. So this is our somatic cell. We have our 2 sets of chromosomes, or 4 chromosomes total. This cell is diploid, meaning that it has both chromosomes in every pair. So we have those two pairs, remember, we have 4 chromosomes total. In one pair, our smaller pair, we have one from dad, one from mom. In our second pair, our larger pair, we have one from dad, one from mom. So this is a diploid cell because it has both chromosomes from mom and dad in it. So this cell is going to undergo meiosis in order to form either sperm or egg cells. So eventually this one cell is going to split so that we have 4 haploid cells. And we're going to look at how that happens. So this is our second step. I included the picture from the step before, but you need to add this second picture now. And I would just draw it like you see up here. I would draw a little arrow from the picture that you drew so that you can draw this next step of meiosis. So, similar to mitosis, cells go through phases G1, S, and G2 before going through meiosis. During the S phase, each chromosome creates a copy of itself. These duplicated chromosomes are known as sister chromatids. Fused sister chromatids resemble the shape of the letter X and together are called homologous chromosomes. So we have our sister chromatids. Each one of the X's you see on this picture is going to be a sister chromatid. So we have sister chromatid, small sister chromatid from dad, which is the dark blue, small sister chromatid from mom, which is the green, large sister chromatid from dad, which is the larger X, the dark blue X, and then large sister chromatid from mom, which is the the larger green X. So we still have our 2 sets of chromosomes, but now the only difference, the only thing that we have happened is we've doubled each one of those chromosomes in there. So we made an identical copy. So we started out with 1 small blue one, now we have 2 small blue ones that make an X. We started out with 1 small green one from mom, now we have 2 small green ones from mom that make an X. We started out with 1 large blue one from dad, now we have 2 large blue chromosomes from dad that make an X. We started out with 1 large green chromosome from mom, now we have 2 large green chromosomes from mom that make an X. So we would say that we have a total of 8 chromosomes in this cell. All we've done is doubled our DNA. And the reason we've doubled our DNA, remember, is because our cells are going to split. So in order for our cells to split, we have to make sure that, sorry about that, in order for our cells to split, we have to make sure that we have the correct number of DNA. So we are going to double our DNA so that at the end we get our haploid cells. So I would draw this picture, I would write these notes down, so that way you have them to reference back to. Next we have step 3 of meiosis. So during this step, we need to add in this little picture. Again, I don't know why there's a little black dot next to it. That does not matter. What we are looking at is the cell, this circle here. So after DNA has been replicated, we went from 4 chromosomes to 8 chromosomes. We just made copies of every single one of those chromosomes. After DNA has been replicated, the like homologous chromosomes line up next to one another. Here, the pairs overlap, exchanging part of a chromatid, resulting in a new combination of DNA. This is called crossing over. So during this time, one of the things that I did not mention in the last slide was homologous chromosomes. So the homologous chromosomes are going to be the 2 small ones and the 2 large ones. So these chromosomes, you get 1 from mom, 1 from dad. They code for the exact same thing. So for example, your eye color. I have brown eyes. I got 1 set of chromosomes from my mom that would code for my eye color, and I got 1 set of chromosomes from my dad that would code for my eye color. Because of the dominant versus recessive, so my dad has brown eyes, brown overpower the green eyes that my mom have, so I have brown eyes. However, I still have the genes for having green eyes, that's just not the color that I have. I have brown because my dad has brown. So every person is like this. You get 1 set of chromosomes that code for a certain thing from mom, 1 set of chromosomes that code for a certain thing from dad. Both, let's say these small ones are what code for eye color. Both of those things code for eye color. And so you have both 1 from mom, 1 from dad. Because they both code for eye color, we would call them homologous chromosomes. They code for the exact same thing. You just get 1 from mom, 1 from dad. So these large ones, maybe those code for height. So you get 1 set of chromosomes from mom, you get 1 set of chromosomes from dad that say you're going to be this height or you're going to be this height. They code for the exact same thing. So these homologous chromosomes are going to line up next to each other, and I have another picture over here We have this light blue one and we have this purple one. They're going to kind of overlap. So they overlap and they're swapping some of their DNA, which we call crossing over. And this is going to be really important in our next set of notes. It's going to lead to why siblings look different, or why everybody looks a little bit different from each other. Nobody has the exact same DNA because of this crossing over. But the important thing to note here is that this is when this occurs in our step 3 of meiosis, before our sister chromatids have split up. So now we're on our step 4. This is where we have our first cell division. So now we went from one cell with a total of 8 chromosomes, 2 sister chromatid pairs. We have split up. Now we have 2 different cells. So we can see from these 2 different cells, we have one large chromosome from mom, we have one small chromosome from dad. And then in the next one, we have one large chromosome from dad, one small chromosome from mom. The big thing to note here is that this is not always how they... The big thing to note here is that each one of these cells is going to get a set of sister chromatids. So we have to have one large one small one large one small one in each cell, because we started out with 2 large ones, 2 small ones in our cell. Now we're going to go from one large one. Now we have one large one small one large one small one. So next, the cell divides. This results in 2 daughter cells that have the same amount of chromosomes but a different combination than the original diploid cell. So if we look at this very first cell, we had one small one from dad, one small one from mom, one large one from dad, one large one from mom. We have the same exact numbers here, 4 chromosomes total in each of these cells, but they have a different combination. And also we can see that they are still in their X's, so we still have our sister chromatids matched together. So again, that's just our identical copy. We have this large one from mom. All we did was made a copy of it, which is why it's an X. We have this small one from dad. All we did was make a copy of it, which is why it's an X. So this is our next step. We have 2 more steps to go. We have step 5 in meiosis, so we need to add in this new picture. And I'll add in here too. If I'm going too fast, pause the video. If you need to rewatch some parts, rewatch those parts. I suggest writing things down and then listening to the video, because then you're not trying to write things down and listen at the same time, and you're not going to get confused. So our step 5, before the cells divide again, the homologous chromosomes line up in the middle of the cell in order to correctly separate during the second cell division. So we can see here that they're not just kind of off to the side. The X's are right on top of each other. They're lined up in the middle. So that way when those 2 cells get pulled apart and we eventually make 4 cells, we get the correct number of DNA in each one of our cells. So I'd add that picture in. I would add this I would add this description of what's going on as well. And now we are in our last step. So lastly, the chromosomes split into chromatids and the cell divides, resulting in 4 new daughter cells. These daughter cells are haploid cells, meaning that they have half the number of chromosomes as the original cell. These haploid daughter cells are also known as gametes. In male animals, these cells are called sperm cells. In female animals, these are called egg cells. So now we are at the very last step of meiosis. We have gone from 2 cells to 4 cells. So if we notice in these 4 cells, we have one large one and one small one in each of these cells. So one large chromosome and one small chromosome. If we look at our original cell, we had 2 small ones and 2 large ones. So now we have what we call haploid cells. So each one of these cells has half of the number as the original cell. So we started out with 4 chromosomes total up here. Now we have 2 chromosomes in each of these cells. And again, I'll come back to why that is. So when when sperm and egg cell go to combine, if we had the total number of cells, we would have way too many chromosomes. So let's say we would have 4 cells in each one of our sperm and egg cells. That's not going to be right, because we want to have 4 cells total. But if we have a cell that has 4 and another cell that has 4, when they combine together, they're going to have 8. So we need to have haploid cells. We have to have cells that have 2 chromosomes and 2 chromosomes, so that when they combine, they get the total of 4 chromosomes, which is what we want. In humans, this number is not 4. Remember, this number is 46. So in humans, when our cells divide, we start with a cell that has 46 and we end with 4 cells. Each one of those 4 cells is going to have 23 chromosomes each. So each one of those 4 cells will have 23 chromosomes. So that way, when the sperm cell that has 23 and the egg cell that has 23 combine, we'll get a total of 46 chromosomes. So all of this was a ton of information. This process is kind of complicated. There is a ton of vocabulary words that you have to know and understand to be able to kind of understand what's going on. So now if you're very confused, this would be a really good time to go back and watch that Amoeba Sisters video of meiosis, so you can kind of get another version or another recap of what's going on. Then we need to talk about next steps. So you need to go take the Target 3A vocabulary practice. Like I said, this is a ton of vocabulary stuff all at once. So go to Canvas, take the vocabulary practice. You have to get 100% on this. This is so that you really understand what's going on with this vocab, because if you don't understand the vocab, you're really not going to understand what's happening in this target, because it's so vocab focused. The next thing you're going to do is Target 3A meiosis reading. So that's just another exposure for what's happening in meiosis. You'll have some questions to answer. That's so that you can get some more practice answering these questions about meiosis before you go and take the test. Then you have the 3A mitosis versus meiosis reading. So again, that's another exposure. We're comparing and contrasting mitosis and meiosis so that we really start to understand the difference between those two. And then you'll have your last set of notes before your assessment.